This is a video tutorial for Curtin, the proteomic data analysis tool brought to you by researchers from the MRC Protein Phosphorylation and Ubiquitination Unit at the University of Dundee. This project is supported by the Aligning Science Across Parkinson's Research Network. Hello Curtin user and welcome to this tutorial, which will show you how to learn about structure, interactors and biology of your experimental hits using Curtin. So after either submitting your own data or clicking on a link, if you scroll down to the volcano plot and select a protein, so for example we'll select ppm1H, it will by default show the raw data distribution first, but you will see that there are some other tabs that we can look at. So let's select protein information to start. So here you will see a brief functional description of your protein and a functional domain structure, both from UniProt. And there is also information about any known mutants of that protein. You can also look at the Proteomics DB database expression profile, which just shows the normalized relative expression of your protein in a variety of tissue types, or also different cell lines. If you want to know about interactors of your protein, you can go to the String DB tab. So this shows you interactors of your protein, and the colour of the text tells you whether the interactor was identified in your dataset or not. So if it's in pink, which all of these proteins are except for ppm1J, which is in grey, so the pink ones were all detected, but this grey one was not. Also, if any of these proteins identified in the dataset pass the log2 fold change cutoff, so the default is a 1.5 fold change, but if you've changed it in the volcano plot settings, then that will also apply here. But if it's past that cutoff, it will be in red if it's increased above 1.5 times the level in the experimental condition you're comparing, or if it's decreased, then it will be in blue. So you can see that ppm1H is in blue because that is decreased beyond 1.5 fold change. And the colours of the lines linking the proteins indicate the type of evidence the association is based on. So a purple line tells you that the link is based on experimental evidence. So all of these links are in purple, but you can see that these three proteins are also linked by a blue line and a green line. So the blue line indicates database evidence and the green line indicates neighbourhood evidence. So the more lines linking two proteins, that indicates a more substantial evidence base. Now there isn't an Interactome Atlas entry for ppm1H, so I'm going to search for a different protein to illustrate this, Rab10. Scroll down and click on Interactome Atlas. So, like with string, this uses colour to show you if the protein was found in your dataset. So here, if the protein is in black, if the actual background is in black this time rather than the text, that indicates that your protein was found in the dataset but was not changed. So the fold change was not greater than 1.5. We don't have any proteins that weren't detected here, but any that are associated with Rab10 or your protein of interest but weren't detected, they would be in teal. And then, as before, red or blue indicates increased or decreased abundance greater than fold change 1.5 or whatever you've specified in the volcano plot settings. And then, also as with string but with a different colour code, the colour of the links between the proteins indicates the evidence base for the association. So if there's a blue link, then that's supported by the HI union. If there's a lilac association, then that's just literature based, and then a peach association indicates both HI union and literature. So as well as looking at interactors of your protein, you can also look at the AlphaFold predicted structure of your protein by just clicking here. So we'll look at the AlphaFold predicted structure for ppm1H. So you'll see, in addition to the structure that appears, you also have the amino acid sequence at the top, and you'll see that when you hover over a particular amino acid in the sequence, you can see it appear highlighted in pink, both in the structure and in the amino acid sequence at the top, which is quite helpful for keeping track of where in the protein you are. So I hope that this video has highlighted how quick and easy Curtin makes it to learn about the biology of your experimental hits by linking all these great tools, so proteomics database, string database, Interactome Atlas, UniProt and AlphaFold structure, all in one place. Thank you very much for watching this tutorial, and please look out for other videos in this series.